Hello everyone, here I'm glad to present some illustrations on my paper, Generalist Equivariant Transformer towards 3D molecular interaction learning in this video. I'm going to first go through some backgrounds and motivations, then demonstrate the details of the proposed method. Then we are going to explore the results of the experiments, and finally I would like to talk about the possible future direction for the research. Now let's go through the background. The paradigm of using deep learning to solve a problem includes collecting data, constructing a model, and optimize the parameters to minimize the objective. And we all know that data matters the most. Data with higher quality and larger scale leads to better generalization of our deep learning models. However, in the realm of structure-based molecular interaction learning, the scale of data is very limited because the structures in the microcosmos is hard to obtain. Especially when we are talking about predicting the binding affinity of two molecules, the labels are hard to get, not only due to the costly wet lab experiments, but also the difficulties in finding new binding pairs. We can see that if we want to predict the affinity of nucleic acids and the small molecules, there's only 149 data publicly available. Instead of waiting for the data to grow, which is actually very slow, we would like to explore whether we can transfer knowledge between different domains to better utilize all data available. Here are the most frequently seen molecules in biology. Each type of molecule uses different building blocks. For example, proteins are built on amino acids, small molecules are built on atoms, nucleic acids are built on nucleotides. However, the basic units are always atoms, and the essence of molecular interactions is secondary bonding between atoms. For example, the hydrogen bond and the pi-pi interactions, and etc., which is regardless of the building blocks. So here comes the question. Can we unify their representations for deep learning to utilize all data from all domains? Intuitively, we can use atomic graphs. However, this makes some large biomolecules too complicated, for example, the proteins. And it also discards the heuristics of the building blocks, or in other words, the inductive bias of different molecular types. If we purely treat each building block as one node in the 3D graph, it lacks atomic information. So is it possible to maintain both the atom level instances and the heuristics of building blocks? We actually propose a unified representation and a corresponding model to process it in the paper. What we do is to treat the molecules as geometric graph of sets, where each node contains a set of atoms. In this way, we can inject the inductive bias of building blocks into the representation. For example, in proteins, each node or block is a residue containing the set of its atoms. The definition of blocks can be arbitrarily adjusted according to your needs, which we will also show in the later experiments. However, everything comes with a price. When we are using the geometric graph of sets for representation, there are three challenges. First, the node features and coordinates are not vectors anymore. They are matrices with variable number of rows, because in different blocks there are different number of atoms. Second, we still want to achieve the E3 equivariance because we're doing structural learning in the 3D space. Moreover, intra-block permutation invariance is also important, since the atoms within a block are unordered. It's a set, remember? To address these challenges, we further propose a generalist transformer to process a unified representation, which includes bilevel attention, feed-forward network, and layer normalization. First is the attention module. Given two blocks I and J, since their features are matrices, we first derive their matrix form queries, keys, and values with linear transformation. We also calculate the relative coordinates between atom pairs in these two blocks, which is further processed into their pairwise distance, that is the Dij. Then in the atom level, relation vector between atom P in block I and atom Q in block J can be obtained from an MLP with their query, key, and distance as inputs. These relation vectors are later transformed and fed into softmax to derive atom-level attention weights. In the block level, relation vector between block I and J is derived by pooling the relation vectors between atom pairs. And then softmax on the block level is implemented to obtain the block level attention weights. For updating the node features and the coordinates, atom level messages for block J to atom P in block I are first obtained with the attention-based weighted sum in the atom level. Then block level attention is used for summing all messages from neighboring blocks. So you can see here the beta ij is the block level attention, and the alpha ij is the atom level weights. The feedboard module is more like local adjustments within each block. We first extract the centroids of the features and the coordinates in the set of atoms, as well as their distances r to the centroids. Then the features and the coordinates of each atom are updated with residual connections with messages from the centroids. The layer normalization is also extended to coordinates, except that the expectation of the projected distribution should be fixed as the same as the original one to maintain equivariance. Next, we arrive at the experimental results. First, we conduct experiments on protein-protein affinity to validate whether the unified representation can perform better in single domains than the naive unified representations. We compared our model with a bunch of baselines, which is very diversified, from invariant GNNs like SNET to state-of-the-art models like Maze, Equiformer, and LeftNet. The test set is split into rigid, medium, and flexible to indicate the extent of conformation change upon binding. So actually, the flexible split is the most difficult one. The experiment is parallelly run for 3 times to achieve a robust evaluation. For different combination of models and representations, our model achieved the best in all splits. On the most difficult split, our model still achieved significant improvements over the baselines. Next is the protein ligand affinity prediction. Our model also obtained satisfactory performance. Here, get PS actually illustrate the flexibility in defining blocks. In this model, we use the fragment-based molecular tokenizations called principal subgraphs to define the blocks. As you can see, it is the fragment-based molecular representation. We can see that the performance is enhanced a lot with better tokenization methods. Then we conduct analysis to answer the following question. Can we really transfer knowledge between different domains? First, we mix the training sets of protein-protein affinity and protein-ligand affinity to see how the performance changed compared to the models trained on single domains. We can see that the performance on both domains improves a little after mixed training. This actually already validates our motivation that the underlying molecular interaction patterns can be shared across different domains. Therefore, the mixed training introduces benefits instead of damages on the performance. We further mixed all available affinity data between different types of molecules for training, except the nucleic acid ligand affinity data, which is actually used as zero-shot test set since it only contains 149 data. It is very inspiring to see that our model achieved moderate correlation in the zero-shot setting, while other baselines all failed. This is a strong evidence that that it is beneficial to mix the training of molecular interactions in different domains, since the underlying physics are shared. Finally, we would like to further discuss some possible directions for future research. We actually can do a lot of things with this unified representation. For example, we can train one pre-trained model for all kinds of biomolecules for structural representations. We can also explore better fragment-based small molecule segmentations, which can be easily integrated into the unified representation just by changing the definition of blocks. Moreover, it is possible to generalize to the non-canonical amino acids by either putting their atoms in the same block or just divide some atoms in the side chains into another block. It is also inspiring to see that recent breakthroughs like AlphaFold 3 and the Rosetta Fold item also adopt a unified model for different molecular interactions. I think unified modeling might be a popular paradigm to address the problem of data scarcity in the future. That's all. Thank you for your attention. Here are the QR codes for our paper and the codes. Thank you for your attention again.